Please don't understand this as a classic research thing. It's not all stuff that I have researched. I have chosen random, hopefully nice examples of what has been done before, just to show a tour d'horizon of what is currently possible with the toolkit that Archaeoscience offers, and how we can sort of squeeze that toolkit a little bit further to get more information about prehistoric temporality. And temporality, of course, not in a sense of how old is something, not in terms of chronology and when were things, and not in terms of are there time axes in prehistory and all the stuff that we have been talking about, but rather to get temporal practice information. How did people actually handle time in past periods? And before I do so, I quickly introduce you with a few slides to the conventional archaeological sources towards or to past temporality. And then we will talk about stable isotopes. We will talk about biomolecules. And we will talk about aDNA, which are the, I think, three key sources that have emerged during the last 2 decades in organic archaeochemistry. There is a large and vast area of inorganic archaeoscience as well, but it's not my field, and it's even more difficult to extract time information with these sources. But I think these organic sources have quite a potential, which I will demonstrate using case studies from human lifetime. So I'm waving a big hello to Irene and her research here, because this is sort of something that could be matching textual evidence if we have matching skeletal finds, for example. I'm going to talk about animal timing. This was my very first talk for Chronoi some years ago, and we've gotten a bit farther in the field so far. And I want to describe what progress I've seen in the last 3 or 4 years. Then I will show you 2 instances how lipids and proteins, which are biomolecules, can help us understand food, of course, because you've mentioned food. And then we will switch to DNA. Summer and winter crops are a topic here. And at that point, I will also shift again to animals. If Glenn Most is here, he will like it, because we had an animal talk last week. I will completely new topic, try to talk with you about the ambling horse and its genetics, and what the genetics of the ambling horse could do for Chronoi. So after this, let's start what archaeology in the beginning, first decades of its history of research had to offer about time and time perception of past people. And of course, we of course, as first thing, we think about calendars. There are a number, or actually a long list of objects that have been tentatively interpreted as prehistoric calendars by several researchers. And you will all know the Goldhut from Schifferstadt and Etzelsdorf and other specimens. If you don't know that object and that kind of object, have a look at the Neues Museum here in Berlin, where it's on display. And I think if it's not been updated, it is there with the calendar interpretation, which has been challenged by a lot of researchers, ranging from statistics to myself to other people. And it's still a debate whether these ornaments on these objects really depict lunisolar systems and calculation systems. And the same is true that all these interpretations are pretty weak and not well grounded, with even older specimens such as Tsokhatsiev's interpretation of this inscribed clay, let's not say tablet because it's not from Mesopotamia, but it's an object made from clay, baked clay from Neolithic Bulgaria, and it could depict a system of a checkerboard pattern, just a pattern, but he also interpreted this in terms of a calendar system, and these are all super problematic. So leaving this field, we come to the fantastic pictures from Chateau Hill, a place where I've been excavating, but not on the East Mount, which is the famous one where these pictures come from. And these have been unearthed by James Mallard in the 1950s. These pictures are super interesting because they are layered. They are Neolithic wall paintings inside domestic houses. And in many instances, these pictures have been repainted and repainted over the course of the time. And while Mallard has, in the 1950s, realized that they have been repainted, there was no instruments to really get closer into how often and so on. And this has been done by the new excavation project by Ian Hodder. And you see paintings that have been unearthed in the new excavations here on the right side. Unfortunately, they are not figurative, but more pattern-like. This would be an example. And on the right side, you see a little thin section into this painting with all the older layers of older paintings behind what we see today, which was the last stage that has been excavated. And of course, it's not possible to uncover each and every layer of these paintings, but it's possible to count them. And there is a very famous article by Nicole Boyven, where she compared painting activities, repainting, replastering of the walls with similar cycles in rural Rajasthan in India, suggesting that maybe life events in the household, like marriages, births, and deaths, could be related to these repainting and replastering events, which is again connecting the house to the life course of its inhabitants. But of course, we only have these tools to establish, using this microstratigraphy approach, to establish some kind of relative chronology, which layer was first and which one was later. But there's no means we can say, okay, this was redone yearly or only at big occasions like weddings or so on, because we lack that precision in all means of dating, like carbon-14 that we have at hand so far. But maybe in the future we will get more tools for this tool, who knows. And another approach to time has traditionally been archaeobotany and archaeozoology, because from the finds you get, like ripe berries that survived in the archaeological record or charred cereals, you can deduce the harvest periods, you can deduce lambing seasons from animal remains. If you have young animals that have been slaughtered, you can estimate, okay, usually they are born in spring, for example, in traditional farming, and then you can assume that, for instance, like in this graph, the early spring in the year of Chateau Hill is the time of sheep birthing, where people were quite busy caring for their flocks, whereas then they would take them in summer to the pastures away from the site in the wider landscape, and where they are also entering their rutting period. And then you can expect new sheep by next spring, and the cycle repeats itself. But this was all inferred from analogy with recent husbandry practices that we have in Anatolia in this case, and by comparison. But there was not really a way to detect whether sheep were really birthing in spring, because who knows? If we look at Central European sheep, we have usually very early spring, if not late winter birthing, and it varies from region to region. And it's also possible that the regime of wild animals has been quite different. And in all these things, especially in these birthing and seasonality things, new Archaeoscience sources have brought in a wealth of new information, and I'm going to show you through this in a minute. And if traditional archaeology wanted to look at human lifetime, there was osteology where we could look at. But the huge problem with human osteology is that humans, fortunately for us, can or actually always remodel their bone. Bone is healing, and events like trauma and malnutrition that leaves a mark in the bone get eventually erased again by healing processes. So it's pretty hard to detect any lifetime events, not even birth from a female skeleton. But the only tissue that is not remodeled that can be looked at are teeth. And teeth have been a key element in this since the beginning of traditional osteology, because, for instance, animal hyperplasia that you can see on the photograph here, a condition that is that occurs if you have a period of severe malnutrition or disease that disturbs your metabolism, results in a lack of buildup of animal, and you see these striations here, and you can of course deduce from the toothing scheme that dentists use in order to say how old a child is, at what time this malnutrition period or episode happened, because we know that the crown of incisors are formed in early childhood, and you can say, okay, here in early childhood there have been some malnutrition or disease episodes. So teeth remain a key issue also with the new sources. If, for instance, if we want to look at migration in prehistory and in history, so if we want to look at if during a lifetime an individual has relocated, and as a maybe not the best, but very illustrative, because it's debated, is example I've chosen Echtwet, which is a female burial of a young adult female that was inhumated in the late Bronze Age of Jutland. And I have given the map here because the first publication assumed that the values that they got out of the tissue from this person point towards southern Germany as the area of origin. But it's just to show you how the principle works. This mummy from a bog was sampled not only for the teeth as a signal for childhood, but also for more recent tissues like the fingernails, as one would also do in forensics. And some of the clothing that survived was also sampled. And it was sampled for a specific isotope, strontium, and its ratio, which is very significant of the region where an individual lives. And during the formation of tissue, constantly the specific strontium signature of the surrounding area is via the food and intake and water incorporated into the respective tissue. So tooth should represent the area where one grew up, whereas later hair and fingernails should represent the area where the person lived short time before the death. And you see that the surrounding soil around this burial has quite a different isotopic signature from the tooth where this person grew up, and also a different signature from other tissues that have been sampled. So the researchers deduced that the Echtwet girl, so quote unquote, actually a young female, because she was buried with a child, likely her own child, that she traveled a lot before she died in Jutland. And the debate is still ongoing whether something is wrong with the signal from the soil and whether this really means that she has been from southern Germany, modern southern Germany, or whether actually the values corroborate origin also in Scandinavia. This is ongoing, but I think it's a very nice example to illustrate the principle, but just remember that, of course, soft tissue such as fingernails and hair does not survive if you have a normal mineral soil burial. Usually we only have the skeleton. So we just compare the local soil with the teeth, and if that's different, then we have to think about a migration during the lifetime. And of course, one could do this in a micro approach across different teeth and find an individual migration history for the individual. So the principle is that we need a trait or a measurement, a value, plus a unremodeled ontogenetically datable tissue, and if we have both of them together, then we get something that is a biographical signal. And we have done this. My PhD student at that time of the Eminöter group that Christoph Markschies has mentioned, Alisa Hujic, we have sampled different teeth of the same individual sequentially for stable isotopes that function as dietary markers, and these are nitrogen delta 15N here and carbon delta 13C, and usually in combined interpretation they can give us some idea about diet. And the samples have come from the linear ceramics cemeteries of Stuttgart-Mühlhausen and Schwetzingen, which are both in Baden-Württemberg, so in southern Germany, and these are the first farmers in Central Europe in the second half of the 6th millennium. And their diets are plotted here. The blue ones are the male individuals, the blue ones are the females, and the green ones are the males. And during their childhood, which is the first molar signal, and during their later childhood, which is the second molar signal, so going towards the end of my own jaw here, we have a pretty similar signal for both sexes. But then there's this interesting peak for males in the late juvenile period, when the last molar, the so-called Weisheitszahn in German, is formed. So this is sort of around age 13, 14. So before the actual eruption, of course, the tooth is formed. And we are still not sure whether this really can be interpreted as a much, much better diet for juvenile males than for juvenile females. But it might actually, if it's really true, and we are still discussing this in the paper draft that we are currently writing together with Corinna Knipper and other researchers, we are currently discussing this, and it could be a hint at a lifetime sort of diet change for young men. Think about that they had a period of hunting away from the settlement or herding animals, maybe a special time in the life of young males before they got really men, or during their adolescence. Lifetime also starts much earlier than the M1, of course, and it's also possible from teeth to find out when a child entered or left infancy stage, where it was still breastfed, and entered childhood. So we don't only see the maturation process, but also the becoming a child process. And here are two sites from Neolithic Anatolia. One is Antike-Kolleg and the other one is Chronoi. And they show an interesting difference in the age when children were weaned. The logic behind this is that delta 15N is a marker for protein. So higher delta 15N values show or hint at a higher share of protein, especially animal protein, in the diet. And of course, breast milk is also on a higher trophic level than the person itself. So milk is always higher in trophic level than the animal the milk comes from, and the same is true for human breast milk. So every suckling baby, no matter if it's an animal or a human, is on a high trophic level and looks very carnivore in a way. And levels drop when more adult food, which is usually on a lower trophic level because plant food is also in it, is introduced. And this happened in one side around age 3, and on here and on the other side it happened a bit earlier, which shows that even in geographically and temporally quite close areas and sites, there were differing practices when children should be weaned from their mother's breast milk. Giving us an idea about how infancy and ages were constructed even in the 6th or even 8th or 9th millennium, like in Neolithic Anatolia. There is a trend in weaning ages. You see here a compilation of weaning ages from skeletal material from Mediterranean and Europe. And on the left side are Neolithic examples, including the 2 we have just seen. And there is, across the late Bronze and Iron Age that you see on the right, we have some declining trend that weaning age gets earlier, so that by the age 3, usually children were weaned. But now there's a big but. These values, delta 15N, 13.3, 13C, cannot distinguish what kind of milk we are seeing in the signal. So if you see this rise in weaning age again in the continuation of the Neolithic, we have to always consider that this could be the onset of the use of animal milk, which is certainly an early Neolithic innovation, but we are not sure yet whether it is something that happened before 7000 BC, when it's first attested in, when milk residues are first attested in vessels, or whether this is a bit later in the process of Neolithization. So watch out, this could be animals' milk that is used as a weaning food, and that looks to us like a very long period of breastfeeding, but actually it's someone else's breast or udder that's used. And here we come to the animals that produce the milk. Of course, we can also from these animals check whether they themselves were weaning their animals. And there's evidence from prehistory that domestic animals were systematically weaned a bit earlier than one would expect looking at wild animals. For themselves, rather than allowing the calf or the sheep to continue suckling. And this you can see here in the delta 15 values and delta from sheep, no, in this case cattle teeth from prehistoric France. So the eigenzeit of animals such as the goat, for instance, is a cycle across the year that starts with the birthing period and then comes the lactation period. And when the young animal is weaned, which naturally happens after 150 days, whereas in a domestic context, you would remove the calf or the kid a bit earlier from the mother, then the rutting period is then again hopefully followed by another gestation, and the next cycle starts. And humans have a way to extend this lactation period by, no matter if they remove the calf or kid or not, by just keep on milking by hand or replace actually the suckling young animal by the milking hand of the human, and to get more milk out of that animal and extend lactation into the next gestation of the animal, like 350 days as a maximum. So that would be a way to get milk for babies if you don't want to breastfeed them anymore. But the birthing season is a big problem here, because even if you extend the lactation that long, you have a time gap where you will not get milk from these animals because they get dry after some time into gestation for hormonal reasons. So in order to get a milk flow around the year for every time you have a baby that needs milk, you have to make more tricks. And these tricks have to do with manipulating the birthing season. And when I gave a first talk on the manipulation of birthing seasons in, I think, 2018 on the Sommerfest or something, wasn't it? Then one of the results was that recent research has shown that the first out-of-season lamps are only as late as the 5th or 4th millennium BCE. So that's Copper Age, so pretty late prehistory. But now evidence is there that we have these in the 6th millennium already. And the principle that's at work here, you take a sheep tooth, a permanent tooth, and you sample a series of samples and take an oxygen isotope out of these samples sequentially from the crown down to the animal border that you see here. And this is the first in time, so here, and this is the last in time, so would be here. And if you get enough samples, then you get a curve. And from this curve, you can say, okay, if the curve is peaking, according to the logic of delta 18O isotopy through the year, this is the summer, and the lowest is the winter. And then here at the very start of your sampling, that should be very close to the birth of that animal. So we can deduce here that this animal was born in something like late summer to autumn. And that's what researchers who have looked at early Neolithic 6th millennium birthing seasons from the Gulf of Marseille, so in southern France, would call out of season lamps, because a late summer birthing is something that does not really fit the logic of the year in a temperate Europe. But not knowing what the actual birthing season in wild animals is, we have a bit of a problem of interpretation here, if that is a result of a domestication and manipulation of timing of the animals and a human interference, or whether this is a relapse of a wild pattern, maybe. But in any case, the diversity of the birthing seasons at these two Neolithic sites, ranging from late winter to early autumn, shows us that something is happening that's not normal to the monoseasonal wild pattern that we have to expect. And we have discussed several of these questions, including this oddity that goats and sheep are expected to have their wild birth season in June and July due to the homeland and where it is not. Over a session organized at the EAA in Kiel, or actually virtually this year, co-organized with Ferran Antolin from the German Archaeological Institute in Berlin and David Orton from York. And we've collected a number of very interesting papers on this topic. And we hope that the recording will soon be published and available. So I will send you the link as soon as I get it. And we are planning a follow-up workshop to continue the discussion on these topics, hopefully within Chronoi. So if the milk is out of the udder, then it lands into a vessel or goes into a vessel, and it's very exciting research by Reihe Salisbury and others, research group around Reihe Salisbury in Wien, have found out that spouted vessels that have for a long time been suspected to be feeding vessels for little children actually indeed contain biomolecules, and we are now at the biomolecule part of the talk, of milk. They have lipids in them that say
they are definitely from milk. We don't know from which species, and strongly suggest, because they were buried together with dead infants in graves, that they were used for feeding the infants. And I'm very curious whether soon research will also show similar evidence for spouted vessels in the Neolithic, because the Neolithic is the phase where we expect the onset of milk use also in infant feeding, of course. And that's super exciting. Lipids are one of the best researched biomolecules we have. But lipids are sort of target analysis. You have to really say, I'm going to look for milk lipids, I'm going to look for honey, for instance, but you cannot just use a shotgun approach and look for what is there. But you can look for what is there if you go for proteins. This is a novel approach, has been forwarded by Tina Warriner and Jessica Handy from York at that time. And usually proteomics are done on dental calculus. You know, if you don't brush your teeth long enough, you get these very hardy concretions on your teeth. But it can also be done on other calcitic deposits that work maybe similar to a dental calculus. And together with Jessica Handy, we were successful to extract proteins trapped in calcitic deposits, let's call it limescale, like in our water castles, from Chateau Hill West, which is my excavation next to the famous East Mount of the site. And here you see a variety of food all together in the same pot. So these pots that we sampled here had dairy from goats, then it had meat from deer and from sheep, together with several pulses and cereals. And we don't know if that's cooking events or if that has been one large pot au feu kind of dish that is represented in this evidence, because we can't micro-excavate these deposits at the time. But it gives us exciting insights, like, for example, pulses. They take some while to be cooked. And we can, with this,
bit irritated because my camera goes on and off, but here it's again there. But we can already from these results deduce that there have been changes in cooking habits at Chateau Hill over the course of its existence, because in the early levels, when we have only simple early Neolithic containers, these containers would not be put into embers. You would not never put them into embers. You would cook in them using either hot stones or hot clay balls, as has been suggested by the research team in Chateau Hill. And then when pots become more elaborate in the second half of the 7th millennium and shapes become more varied, it's possible to put these into embers because the fabrics also are advanced and have a better temper in them. And the end of this development is the pot stand that we see at the very end of the Chateau Hill East sequence, and in very refined and nice form at our West Mount in the 6th millennium, and a way of putting the pot on top of the embers and have a steady heat addition to your meal. And that is something that goes with a lot of dairy cooking practices, with fermentation, with cooking pulses for a long time. So we see a fast food on the left side, kind of shock cooking with hot stones or hot clay balls, and a gradual, or maybe not so gradual, transition to a slow simmering and slow cooking process. And that tells us a lot of how time was organized in the households, and how time was diverted to food preparation rather than other tasks. Coming to genetics, finally, we can look at plants. And plants, as we know them today, cereals especially, have different timing patterns. You can either sow them in the autumn and leave them through the winter, where they receive a shock called vernalization, which in the cold, which fosters in the spring the germination and the reproductive cycle of the cereals to unfold. Whereas spring zone cereals can do without this vernalization pause during the winter and ripen until summer, and you can just create within one very short season your cereals for your harvest. And whether cereals follow one or the other pattern, this is a genetic thing which has been identified using modern land races, but also modern industrial crops. It's certain genes that distinguish winter and summer crops. But at the moment, we can only say, okay, a pattern it's a genetic pattern, but it's not possible to really trace these genes in prehistoric plant remains due to preservation issues, because DNA from plants is really tricky to get out of archaeological finds. So currently we have no idea whether the first summer crops are something that developed when the Neolithic got out of its Near Eastern homeland, where you have natural winter sowing wild cereals, into temperate Europe, where the warm season is so short that you have to find a solution and circumvent the need for vernalization by selecting mutations that can do without vernalization. So this is likely something that happened in prehistory, but we don't have a proof for these different timing regimes in agriculture. And to sum up the main part of my talk, chronobiology in domesticates, but also in humans, is, or especially in domesticates, is a matter not only of genetics, but mostly of the seasonal regime the organism is in, and also of targeted timed husbandry practices. And that together is in its interplay between all the elements of a social economy, plants, animals, housing, other activities, is a fascinating and very entangled topic to maybe investigate in a case study. But now the ambling horse. My last and maybe a bit experimental point that I want to make. We start with depictions of galloping horses. And for over, I'd say almost 20,000 years, humans have depicted galloping horses like this, with a flying horse that has both or all four legs stretched out. And this is entirely wrong. A horse does not gallop like this. But horses gallop so fast that we can't, with our naked eye, we can't really see what's happening. So that was the idea that humans had about human gallop from the Paleolithic onwards. So that painting is from Lascaux, the famous galloping horse, and the other one is just some derby from 1821 in England. And that has been a debate in the 19th century whether this is really true. There have been people who argued, okay, maybe I'm mistaken, but sometimes I get a glimpse if I look at a galloping horse that this might not be reality. And a rider and horse owner hired actually a photographer, a pioneer of photography, Edward Muybridge. You see his life dates and his portrait here, to make sequential photos. And this was really a feat for early photography, to make sequential stills of horses in motion to resolve this issue. And that's the famous sequence. It's not the photos actually, but the somewhat rendered version for graphic clarity, that made the point and proved that galloping horses fly when they have their feet together like this. So that's the flying phase in the gallop, whereas when they have their legs stretched out, one foot is touching the ground. So this was resolved in 1872. This was when we understood how a galloping horse really puts the feet on the ground. And reading this, it was just something that you find on Wikipedia. Reading this, I started to think about the gallop, which is a completely different gait than what we humans can do. This is the photos that Edward Muybridge has done for walking horses. This is a trotting horse, if you arrange these sequential photos like a film. And this is a human walking, and a human running, of course, is also the same. It's a two-beat. Yeah? A trotting horse and a walking horse, it's two-beat, and also a human running or walking is a two-beat. But look at this. The gallop is entirely different. It's a three-beat that even Latin poets have put as quadrupedante putrem sunitu quatit ungula campum. It's a remarkably different thing. And this was something that humans, and here we come to Chronoi issue, first experienced from the late 4th millennium BCE onwards, when they first mounted horses and started to ride during the domestication of the horse. And it must have induced an entirely different feeling of movement and of rhythm. And I wonder whether they had a similar kind of aha moment when they first mounted horses and got into a three-beat, like we had when we looked at the galloping horse pictures for the first time in 1872. So the question that I would leave for the future of Chronoi is, did the inception of riding create new rhythms, maybe in music? Are riding societies more three-beat music societies? Are there relationships between time perception and the riding you have? And as a little addition to this, there's one more gait that not everybody knows about, and that's the pace. And the pace is actually like a camel is walking with both legs on one side moving at the same time, which gives a sort of like on a ship, shaking or a very soft ship feeling. And the pace, interestingly, is genetically determined. You see the name of the gene variant here on the slide. And it's been observed in a number of horses. They cluster, of course, in Iceland, and I'd love to talk with Gavin Lucas next year about this. And research has so far found out that first gaited horses that have this gene that induces the pace rather than the gallop after the trot first appears in the early medieval and Central Europe, and it's thought to be something that actually is related to the Icelandic horses. But looking at Mongolian horses, and that takes me to Bonn again, that I have seen, I'm pretty sure that I remember pacing horses there as well. And the pace is again a very peculiar rhythm of riding and another feeling of time. And I'm very grateful to my lecture, or actually teaching horse, Jufur, that taught me what the pace is and what the tölt is. So to sum up, I think that this is the one instant where we have an archaeo-DNA clue to prehistoric timing and rhythm in the genetics of horses that we could maybe even extract for prehistoric horses. And I'm very much looking forward to seeing more of this research in the near future. So that would be it. And I'm looking forward to your questions.